Hi, thank you for inviting me to Labroots Genetics Virtual Week. My name is Nikita Mehta. Before I begin, I wish to disclose that I'm currently employed at Memorial Sloan Kettering Cancer Center, but the work that I'll be presenting today is from the time when I was employed at the Mayo Clinic. I have no other financial disclosures or conflicts of interest. My goal today is to introduce you to a method of classifying variants identified in hematological malignancies and tumor samples. We'll start by discussing the basis for this method, which includes key elements from professional guidelines. We'll also discuss some of the evidence criteria in more detail to better understand how to perform a variant curation or annotation for molecular profiling of a neoplasm. Finally, we'll go through some curation examples and compare the methodology with other described approaches. To begin, let's take a recent historical look at curation methods. Variant curation is defined as the interpretation of genetic alterations. Because interpretation is inherently subjective, standardization has always been considered important for consistency in classification and subsequently medical care. The first guidelines for the process were released by the American College of Medical Genetics in 2000, which were for the interpretation of constitutional variants. The guidelines have subsequently undergone revisions, with the most recent revision being a joint effort between the ACMG and the Association of Molecular Pathology in 2015. You are all probably familiar with this image from the 2015 guidelines, which summarizes the different types of evidence that should be researched to aid in classification. They also provide rules for how those pieces of evidence can be combined into a classification, namely benign, likely benign, variant of uncertain significance, likely pathogenic, or pathogenic. With advances in the field, next-generation sequencing and more rigorous sequencing technologies have drastically expanded the uses of molecular techniques. Consequently, a need arose for creating a standardized method of interpreting variants acquired in tumors. Because the ACMG AMP 2015 guidelines specifically indicate that, quote, a different set of interpretation guidelines is needed for somatic variants, end quote, AMP created a separate working group to establish these rules. In 2017, AMP, the American Society of Clinical Oncology, and the College of American Pathologists developed a method of tiering variants, primarily based on the availability of therapeutic, prognostic, and/or diagnostic implications for the variant. Then in 2018, a paper came out from the Clinical Genome Resource, or ClinGen for short, who demonstrated that the methods for evidence combination in the ACMG AMP 2015 guidelines generally fits a Bayesian framework, meaning that the described rules for combination from the 2015 guidelines yields probabilities defined for the qualitative classifications. So most of the evidence criteria in the pathogenic section, when combined as seen on the left, yielded a posterior probability of pathogenicity of at least 99%. The likely pathogenic section yielded a posterior probability of pathogenicity of between 90 and 99%. The likely benign section was between 0.1 and 10%, and the benign section was less than 0.1%. It is these three papers that primarily make up the basis for the methodology I want to introduce today. When I was at the Mayo Clinic and working on somatic interpretation of variants, mainly for myeloid malignancy, I quickly realized that the types of evidence I was searching for in the context of Mendelian disease was very similar to cancer. Thus, we adopted the structure of the ACMG AMP 2015 guidelines, which were the only guidelines available at the time, to help categorize the evidence found. When the AMP ASCO CAP 2017 guidelines were released, we found the focus to be on without including the ability to comment on whether the variant drives tumorigenesis. So our team continued to use a modified version of the ACMG AMP guidelines, but started to incorporate the levels of evidence for clinical significance defined in the AMP ASCO CAP guidelines. Our justification for generalizing the ACMG-AMP germline approach finally came with the 2018 ClinGen paper. Of course, the purpose of the ACMG-AMP guidelines couldn't be translated perfectly for use, so we made some changes to fit a somatic workflow. First, we excluded a number of criteria that wouldn't be of use in the somatic or cancer realm. The ones listed here are typically not used, with the most common exception being PP1. If the variant occurs in a gene associated with the germline disorder, and the mechanism of the germline disorder aligns with the mechanism of tumorigenesis, co-segregation data is still likely relevant. This is especially useful for hematological malignancies where discerning between acquired and constitutional variants may be more difficult, as the matched normal sample to separate out germline variants cannot be blood. We also try to provide more specificity in the guidelines for the criteria that did remain. For example, PVS1 rules were made more specific in a recent publication by the ClinGen Sequence Variant Interpretation Group, and we made it mandatory to follow the process set forth by this organization of experts. We also utilized data from the AMP-ASCO-CAP guidelines to direct our minor allele frequency cutoffs for BA1 and BS1. They surveyed laboratories to determine the MAF cutoffs used to establish a variant as being too frequent for disease. Instead of the 1% cutoff that most laboratories were using, we conservatively selected 3% to account for the allele frequency of some heritable pathogenic founder alterations seen in common cancer predisposition genes. We also provided guidance on reliable databases and tools for certain types of evidence. PS4 originally was meant to rely on case control data, but instead we defined how many cases are needed by tumor type and somatic status to use this code at the strong level. We also indicated that case data ideally comes from Cosmic, CBioPortal, and the literature, but any other reliable source is fair. Other codes that suffer from too many choices are PP3 and BP4, which are related to the in silico or computational tools. Again, specific in silico tools were recommended with guidance for cutoffs for missense variants and splicing variants. However, I would note that a year later, there are likely other tools with better performance that should perhaps at least be incorporated into the approach. These will likely remain dynamic as more tools are created. The last major change was made to PM5. While this code was originally meant to be applied when any other missense variant at the same residue as the variant of interest has been classified as pathogenic, we limited the use of the code by asking the curator to verify the significance of the residue or to compare the amino acid properties, structure, and conservation or functionality of the involved residues. Furthermore, mild differences between the involved residues may result in using this code at the supporting level of strength, or not at all. You'll find that there are other differences with this modified version of the guidelines, which are fully described in the text of the cited paper. After you are able to determine which criteria can be applied to your variant, that evidence can be combined into a classification. The rules for combination are largely based on the original table from the ACMG-AMP 2015 guidelines, because again, the structure generally fits a Bayesian framework. However, we made a few minor adjustments and presented the rules combination for the codes only defined in our approach. Sometimes the best way to learn a new method is to go through examples. I've picked a few variants to demonstrate application of the rules, and will compare the classifications with other methods. In the first case, an 80-year-old was diagnosed with myelodysplastic syndrome and underwent NGS to help guide therapy and provide prognostic information. A frameshift duplication in DDX41 was identified in the patient. DDX41 is an RNA helicase that is part of the dead box protein family. Acquired loss-of-function alterations are reported in up to 4% of adults with MDS or AML, but the gene is also associated with a hereditary predisposition to MDS and AML. So the first thing to assess when doing a variant curation is what type of variant is it. A duplication such as the one seen in this patient will shift the reading frame and result in a premature truncation codon. The patient's duplication creates a premature truncation codon in exon 5, which would cause the mRNA to be degraded via nonsense-mediated decay, as it is upstream of the last 50 to 55 nucleotides of the penultimate exon and the last exon itself. As we were curating and reporting in the biologically relevant transcript, PVS1 could be applied at the very strong level of strength because it is highly likely that the variant produced a negligible amount of protein and can thus be considered a null variant. Next, it is best to assess case control data. The variant is present in NOMAD, which is a database that is meant to represent the general healthy population. As you might imagine, excluding patients with later onset disorders can be more difficult, and clonal hematopoiesis is another phenomenon that isn't currently addressed in NOMAD. Regardless, we noticed that the variant was seen in 22 European heterozygotes, which intuitively may feel like a very high frequency. However, the MAF is not high enough to favor neutrality. It has to be over 1% to apply BS1. It is also worth noting that the variant is described in the literature as a germline founder variant in the Caucasian population, so we really shouldn't exclude the variant based on its frequency. Plus, some of the other individuals in NOMAD were relatively young, making it less likely that they would have already presented with an associated phenotype so as to have been excluded from NOMAD in the first place. When digging a little deeper into the literature, we found that there are several families in which the variant segregates with MDS or AML in spite of the lack of somatic cases. I want to take a minute to explain how the calculations in Jarvik and Browning's paper was used here, which we recommend for use in determining PP1 strength level. Using the pedigree of one of the carrier families as an example, note that black circles or squares indicates a diagnosis of MDS or AML, and the stars indicate mutation carriers. First, understanding that hereditary cancer syndromes have reduced penetrance makes the lack of phenotype in the two unaffected brothers less concerning. Next, to calculate the probability that the variant has segregated in the affected family members by chance, you would first assign a value of 1 or 100% to the proband indicated by the arrow. This means you are conservatively assuming that the proband definitely has the DDX41 variant by chance. Then, the other family members receive a 1/2 or 50% chance to have the variant just based on how alleles are passed on for an autosomal dominant condition. That would be 1/2 for the brother and 1/2 for the father, or 1/2 squared. That makes the chance that this variant has segregated with disease in this family by chance 1 times 1/2 times 1/2, or 1/4. For a single family, 1/4 isn't even enough to use PP1 at the supporting level of strength. However, because there were multiple families with this variant, we were able to reach an N value that was less than 1 in 16, which is the threshold for applying PP1 at the strong level of strength. In the final research step, you can assess the variant functionally, whether it is based on actual functional studies or in silico evidence for certain variants. In this case, there was no variant-specific functional data, so PS3 cannot be applied, but there was a study that showed complete loss of DDX41 does result in increased cell proliferation and colony formation, which again supports the application of PVS1. Ultimately, PVS1 and PP1 strong apply to this variant, which it combines to a classification of pathogenic. For this patient, we technically do not know whether this variant is acquired or constitutional, but the variant allele fraction was approximately 50%, and recommendations were made for the patient to confirm germline origin through testing of skin fibroblasts. Because origin is unclear, we still have to assess the variant for possible clinical significance if it is somatic. There are single studies that suggest better response to lenalidomide in those with DDX41 alterations, and also possibly more aggressive disease with reduced survival. However, these findings had not been replicated, so the variant did not even meet evidence level D. Now let's compare this classification to the standard at the time, the AMP-ASCO-CAP 2017 guidelines, as well as a new publication from ClinGen, the Cancer Genomics Consortium, and the Variant Interpretation for Cancer Consortium. AMP-ASCO-CAP guidelines indicate that the variant type does align with Tier 1 or Tier 2, but there are no somatic cases of this variant. Unfortunately, all the tiers 1 through 4, can coincide with germline cases being present or absent, so the germline data doesn't help distinguish the tier. What ultimately helps determine the classification is the lack of consensus around a therapeutic or prognostic effect, which moves the classification to Tier 3. The ClinGen CGC-Vic 2022 guidelines have the same basis as our guidelines, but there are some differences for certain criteria, and a quantitative approach was encouraged. For this example, if it was unknown that DDX41 was associated with a germline predisposition, this variant would meet the OVS1 criterion, which is identical to PVS1, and the variant would still be reported as likely oncogenic. However, these guidelines do also indicate that variants in a gene known to be associated with a hereditary susceptibility to cancer may be classified by ClinGen gene-specific germline rules when they exist, or the ACMG AMP 2015 guidelines. The latter would result in a pathogenic classification like our conclusion. The next variant we'll discuss was identified in a 39-year-old patient who had essential thrombocythemia. In this patient sample, the W515S missense variant was detected in MPL. The MPL gene encodes a receptor for the thrombopoietin ligand and regulates proliferation and differentiation of megakaryocytes and platelets. Somatic alterations are found in about 3 to 5% of ET patients, where MPL generally functions as an oncogene. Similar to tumorigenesis, constitutive activation is also seen in variants that cause autosomal dominant thrombocythemia. However, loss-of-function variants cause autosomal recessive congenital amegakaryocytic thrombocytopenia. The evidence for this variant is substantial. Although this variant is a missense variant, which can be hard to resolve from VUS status, the variant is located in the juxtamembrane domain at a residue that typically blocks receptor dimerization necessary for protein activation. And W515 is a hotspot residue, which includes the pathogenic W515L and W515K variants. This corresponds with PM1 and PM5. Furthermore, the variant has been reported in at least 18 cases of myeloproliferative neoplasm, 3 of which were confirmed to be somatic in origin, and the variant is absent from population databases. The latter corresponds with PM2, but PS4 cannot be applied since there aren't at least 5 cases of MPN with this somatic variant, or 10 confirmed somatic cases in total, including at least 1 MPN. However, there is in vitro and in vivo functional data for this variant that shows that the mutant protein is constitutively active and causes ligand-independent cell growth, even though in silico tools predict a benign effect. When the criteria are combined, the classification is likely pathogenic. However, when the variant was reviewed in our division, we upgraded the variant to pathogenic. Our generally conservative threshold for somatic case counts had the potential to be an issue in hematological malignancy because, again, a matched normal is not as easy as drawing blood. But the overwhelming case count at this hotspot residue was highly suspicious. Furthermore, it was upgraded given the variant meets level A evidence, as MPL alterations, particularly at W515, are a diagnostic marker for MPN. Similarly, AMP-ASCO-CAP guidelines resolved to a Tier 1 classification. The ClinGen CGC-Vic guidelines resolved to a likely oncogenic classification mainly because the cases cannot be counted as stronger evidence due to lack of confirmed somatic status. Please note that different missense predictors are recommended for use in the ClinGen CGC-Vic guidelines, which is why OP1 was applied instead of SBP1, the equivalent of BP4, but this wouldn't affect the likely oncogenic classification either way. The last variant we'll discuss was identified in a 79-year-old patient with acute leukemia. In this patient sample, the G662C missense variant was detected in SMC3. The SMC3 gene encodes a component of the cohesin complex, which has roles in hematopoietic stem cell differentiation, maintenance of chromatin structure and gene expression, and double-strand DNA break repair. Somatic alterations are found in about 1 to 4% of AML and MDS, where variants result in loss of function. Constitutional de novo loss of function variants are also associated with Cornelia de Lange syndrome. Again, we have a missense variant that doesn't typically favor pathogenicity or neutrality, so we'll need to find other evidence that helps move us one way or the other. At the time, I was able to find in the literature that the variant is located at a glycine residue in a hinge region, and that the residue is part of a series of glycine residues that are important for dimerization. Thus, PM1 was applied. When looking at case control data, I was able to find 3 cases of AML with the somatic variant, and an additional AML case where the origin of the variant was unknown. While this is certainly suspicious, it does not meet our conservative threshold for PS4. However, the variant is at least absent from general population databases. Finally, there was a study that showed that the mutant protein decreases erythroid differentiation, which means that PS3 can be applied, although in reporting it should be mentioned that there is only a single functional study. When the criteria are combined, the classification is likely pathogenic. The potential clinical significance of this variant is less certain, though, as there are controversial studies about prognostic implication for variants in this gene, and only one paper that suggests an association with SMC3 variants and secondary AML. In the AMP-ASCO-CAP guidelines, they indicate that variants of uncertain significance will often be missense variants, although Tier 1 and Tier 2 can be associated with loss of function or activating missense variants, as we saw in the previous example. Of course, the somatic cases support Tier 1 or Tier 2 classification too. However, it is again the lack of therapeutic, prognostic, or diagnostic significance that settles the classification to Tier 3. The classification based on the ClinGen CGC-Vic guidelines, on the other hand, will likely align with our classification. Note that one reason it may not align is dependent on whether or not the functional data would be considered sufficient for application of OS2. The functional study does demonstrate loss of a function that is typically associated with the SMC3 protein, but it does not result in increased cell proliferation or tumor formation. If functional data couldn't translate to at least supporting level evidence, even the ability to add an OP3 for the 3 confirmed somatic cases would not lead to a likely oncogenic classification. I hope this has helped to introduce some somatic variant curation methods. I want to thank my co-authors, Dr. Viswanatha and Dr. He, and also thank you for your attention. I'm happy to answer any questions.